Hi, in this lecture we are going to start our study of chapter 23, which will look at nutrition, metabolism, appetite, hunger, body temperature regulation. It's kind of a whole hodgepodge of everything that's thrown in here. So in this first lecture, we're going to look at metabolic pathways, and this is meant to be a review because I know you already learned them. But to begin with, let's define some terms. So when we look at metabolic processes, we have what is called catabolism or catabolic, and this is breaking things down. So when we use our digestive system, we take in food, we break it down mechanically and chemically into small pieces. We take those nutrients and absorb them, send them over to our cells, and our cells are going to continue this process to break down the nutrients and get ATP. Anabolic is building. So the other thing the cells do with all those pieces, those amino acids and glucose and fatty acids, is they use them for anabolic processes. So usually for maintenance or repair. So this is how we can grow. This is important for making secretions, hormones, enzymes. We even have some reserves. What we can store in our body is glycogen and fat. Notice we cannot store protein. We don't have anywhere to put that extra protein. In fact, you'll see shortly, it just gets converted into glycogen and usually fat. And then the nutrient pool is just everything that's available in your body at any given moment for the catabolic and the anabolic reactions. So if you had Bio 119, you would have seen this graphic before. And when we look at aerobic cellular respiration, it's the process of using oxygen. So when we say aerobic, we must use oxygen for this to occur. And it's 4 processes. These are called metabolic pathways, which is just a sequence of events. It's a sequence of very specific steps that occur, and every step has an enzyme. So the first step when we're trying to make ATP through aerobic respiration is something called glycolysis. And notice glycolysis is happening out here in the cytoplasm. Once we go through glycolysis, we then move on to the second step, which is called the transition step. Now, this big purple-pink thing is a mitochondria. So notice that the transition step, the citric acid cycle, and something called OX-phos, these processes all occur in the mitochondria. So the transition step is a short step, and then we go into step 3, which is the citric acid cycle, and this is a cycle that is always running. And then once we process these nutrients through the citric acid cycle, it's sometimes known as the Krebs cycle, we go through the last one, which is called OX-phos. So OX-phos stands for oxidative phosphorylation, and what that tells you is that that has to have oxygen. That's why it's called oxidative phosphorylation. Now, there's 2 parts to this. There's something called an electron transport chain, which does just what it sounds like. It moves electrons through. And then the second piece of this is something called chemiosmosis, which we'll be looking at. So notice that these pathways always occur in the same order. They are there to produce ATP, so we get some ATP in certain pathways. And the goal is to produce enough ATP to sustain life. So glycolysis is always going to be the very first one that happens. So if we're looking at carbohydrate metabolism, we take glucose. Now, carbohydrate metabolism can only burn glucose. It cannot burn fructose nor galactose. Now, we eat those. We eat, fruit sugar. And so those sugars just go to the liver, and the liver will convert them into glucose so we can use them for energy. Now, remember that glucose is stored as glycogen, so glycogen can readily be converted back to glucose. But what we're going to do for this pathway is take glucose. We need the oxygen, which we have a respiratory system for. When it runs through these 4 pathways, we're going to produce ATP, which our cells need for energy, water, and carbon dioxide. And remember, the carbon dioxide leaves the cell, enters the blood, and gets transported up to your lungs so you can exhale it. Now, the first step is called glycolysis, and it happens first no matter what. But what if you don't have a lot of oxygen available? What if you're exercising at such a high rate, like you're running uphill, or you're lifting really heavy weights, or you're jumping rope? So you're working out really, really fast, faster than you can get oxygen to your muscles. So glycolysis can happen with or without basically oxygen because it doesn't need it. And that's why it's always step one to occur. So this process happens every single time. It takes glucose. Its job is to grab a glucose. It converts it into something called pyruvate, or we could say pyruvic acid, and then it's going to kick off a couple end products for us. Because it's so fast, it doesn't need oxygen. And so it's our like when we're very, very high intense activities. A real easy way to tell if you're burning anaerobic, just glycolysis, is if you can talk when you exercise. If you're exercising at such a high intensity that it's hard to talk, you're working just anaerobically with glycolysis, and you won't be able to do it real long. So when we look at glycolysis, this is the long, we're not doing everything here. This might have maybe bad bio 119 flashbacks. So what I want you to know for each pathway is what goes in. The reactant is glucose. Now remember, you do need insulin to get glucose into your cells. And then it goes through a bunch of steps, and it ends up forming pyruvic acid, which you can call pyruvic acid or pyruvate, either's fine with me. We strip off hydrogen. So as we go through, if you look at glucose, notice that glucose is going to have 12 hydrogens. We eat for hydrogen. That's what we're burning. Our goal is to get those hydrogens off. So those hydrogens get stripped off. There's a little, I call it a cellular taxicab. NAD comes from the B vitamin niacin. It strips hydrogens off and forms NADH, and it takes them into the mitochondria for oxphos, which we'll deal with later. But you only get a net of 2 ATP for every glucose. It's not very much ATP. That's why we can't sustain these activities very long. If you think about how long you can jump rope or sprint. So if oxygen is available, this pyruvic acid or pyruvate that is formed is just going to continue into the mitochondria and undergo the transition step, and it's just going to work its way through. Well, what if, again, we're working at such a high, fast level, we just can't keep up. We can't get oxygen to our muscles quickly. Well, if there's no oxygen, this oxphos is going to stop. It won't work. And remember, all these little hydrogens that are being picked up by the little taxicab, the NADH, it's going to pull hydrogen several places. It has to drop all those hydrogens off here so they can be used for this thing called chemiosmosis. Well, if this is shut down and not running, then there's nowhere to drop those hydrogens off. This thing's closed. It's like, nope, can't take them. So what happens is this little NADH, we'll just draw one over here, it wants to dump those off, but it can't. Well, it can't store them. So what it does is it brings them back to pyruvate, and it throws them onto pyruvate. It's like, here, you take them. Well, when pyruvate takes hydrogen, we call it lactate. So we get lactic acid. So when we're working anaerobically, we can only do it for shorter time. One, we start to run out of phosphates, and we make all this lactate or lactic acid because basically the other pathways just shut down. So that's why we form lactic acid. Now, if we have aerobic metabolism, if we have oxygen available, then this pyruvate will simply go on to my next step, which is the transition step. Now, if we look at this overview, the transition step, pyruvate comes in. It's starting with pyruvate, but what's leaving is this thing called acetyl-CoA. So the transition step is just taking a pyruvate and converting it, because the citric acid cycle, it starts with acetyl-CoA. So you need a transition to convert the pyruvate into the acetyl-CoA so it can get inside the citric acid cycle. And that's all it does. So when we're looking at the transition reaction, we have our glycolysis, we get pyruvic acid or pyruvate, and it goes in the mitochondria and it gets converted into acetyl-CoA to continue. Well, when it doesn't, we're not memorizing the steps, but we do get some more hydrogen that we can use later, and we do get one of those CO2s we got to get rid of. So the CO2, remember, just diffuses out, goes up to your lungs, and we exhale it. So now we've got our acetyl-CoA. We can enter our next pathway, and our next pathway is called the citric acid cycle or Krebs cycle. I do want you to know the first step of this because it's pretty important soon. We'll be talking about the significance. So acetyl-CoA is coming in. It combines with something called oxaloacetic acid. See how this is the last one formed? So the two combine and they form citric acid. That's why it's called the citric acid cycle. We are not memorizing the steps. Citric acid goes through many reactions. Notice that we strip a bunch of hydrogen off, which we need over in oxphos. We get an ATP, not very much, but we do form a lot of CO2. So that CO2, again, has to go into the blood where we exhale it. So when we're looking at these pathways and we're looking at respiration, we're getting our CO2 here. These two, transition and citric acid, they're generating that CO2, which we know is a waste product that we have to eliminate. So now we're going to move inside to what's called oxphos, and there's two steps here. There's the electron transport chain and chemiosmosis. Now, before I jump over there, I also want you to just recognize the end products. You don't have to know the numbers, but just know we get some of these NADH, and FADH is another taxi cab. Just think of it that way. So we strip hydrogen, we get a little bit of ATP, and we get CO2. So now we are going to go over inside the mitochondria where we're doing what's called oxphos. So you might remember that the mitochondria has some membranes inside, this inner membrane. These little blue things here, you'll see Roman numerals 1, 2, 3, and 4. So this part right here, these are the yellow phospholipids, this piece of it is called the electron transport chain. So oxphos has two components, electron transport chain, chemiosmosis. And what happens is it needs oxygen. So this will only run if oxygen is available, and it uses hydrogen. So when we had those NADH, we'll just stick with those right now, that comes in and it drops off its hydrogens. And then it goes back out and picks up more. So that's all it's doing. It's going back and forth and back and forth. When it drops off the hydrogens, whether it's from the NADH taxicab or the FADH2, a hydrogen has one proton and one electron. So it's immediately split. The electron, this little red thing, gets shuttled through these little guys called the proteins, the electron transport chain. So it just goes boom, 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 boom. Okay. Now, why is that significant? The electron has energy. And as the electron gets past, think of that game Hot Potato, right? It's just going boom, boom, boom through every one of them, then it pops in. This energy from this electron shuttling is used to pump the protons out. So when we split that hydrogen, we're left with what we call a proton, because one proton, so we say H+. And notice what we do is the energy from the electrons creates a proton gradient. It creates a lot of hydrogens on one side of the membrane and fewer on the other. So I like to think of this as a mini battery. We have a plus side, a positive, and a negative. And if we allow flow, if we allow those protons to come through, we will be able to generate ATP, and that's exactly what's going to happen. Now, real quick, these little proteins are called cytochromes, and there's 4. You don't have to memorize a number, but I do want you to know that some of the components are some minerals. So we have sulfur, iron, and copper. So one reason why we need these in our diet is because they help form the electron transport chain, which is going to shuttle those electrons through. And remember, that generates the energy to pump the hydrogens and make the battery. Well, if anything blocks this chain, and if I can't shuttle electrons through, I can't pump these hydrogens out and make my battery, I'm going to have a problem. Because if I don't have that battery, I'm not going to be able to make ATP, and it's going to kill me. And you'll see here that at certain channels, this is called an ATP synthase. It's basically a little turbine that spins. So whenever we have this complex, those hydrogens come back across, and as they come across, they spin this turbine. And as it's spinning, it makes ATP. And so you have to have oxygen there to catch all those hydrogens and electrons, because otherwise you would change the pH. And we form what's called water, or metabolic water. So chemiosmosis is just the process of using a hydrogen ion gradient to make ATP. And that's why oxphos has the two components, because you have to have the electron transport chain first to make the gradient, and then you have to have the chemiosmosis, when those come back through and spin the little molecular turbines. So two things are going to occur. And then we're going to generate all the ATP. And again, we need the oxygen there to kind of collect, to grab all those hydrogens and electrons, and it forms water. And this is called metabolic water. Now, this is critical for cells. If we lose the ability to make ATP this way, cells die, tissues die, organs die. You go into organ failure. So just know that there's certain chemicals and poisons that interrupt the chain. So for example, you've all heard of cyanide. And what cyanide does is it blocks one of those protein transporters. So it decreases the amount of hydrogen you can pump. Well, if you can't pump the hydrogen, remember, you can't make the battery. And so you can't make ATP. And if you can't make ATP, then death will occur. And that's how that one works. Now, if we take a glucose molecule and we just kind of look at it and look at the ATP, you don't have to memorize the exact number, but it's roughly 32 ATP for one glucose when you generate it aerobically with oxygen. If you do anaerobic, remember, it's only 2. So when we're looking at carbohydrate metabolism, remember that we can store glucose as glycogen, and we can also break it down. We can take glycogen and release it into glucose. You don't have to memorize this glucose 6-phosphate. It's just the intermediary molecule. But what I would like you to know is some terms that kind of a review for you. So glycogenesis, remember, genesis is going to be when we take glucose and we store it as glycogen. So anytime we have extra glucose, our body's really good at storing it, and it's going to store it in our skeletal muscle and our liver. We have a lot more skeletal muscle than liver, so we store a lot more in our muscle than in our liver. Whereas glycogenolysis is the opposite. Now you see the lice. Now you're taking glycogen and you're breaking it back down into glucose. So when you have it and you don't have enough glucose, you can break it out of reserve. It's like your savings of glucose. Now, glycogenolysis happens pretty quick in the liver. You do not have to memorize the enzyme, but the liver has an enzyme that allows it to go really fast. The problem is we only store about 400 kcals worth of glucose, basically glycogen, in our liver. So it'll get you through a couple hours when you get really, really hungry. You might notice you're like really hungry and you're irritable. And then all of a sudden you might feel a little bit better. That's because you are actually breaking down your glycogen from your liver, and it's maintaining your blood glucose levels. And then if you get hungry again, then you're like really hungry and you have to eat because you've used that up. We can take it out of skeletal muscle. Skeletal muscle stores roughly 1,800 kcals worth. So if you add it up, right, you have, again, roughly 2,200 calories worth of glycogen stored if you're eating well. That'll get you through a one-day fast. The muscle can break it down. It just takes a little bit longer. It doesn't have this enzyme. So it actually has to break it down into pyruvic acid and then send that to the liver so the liver can convert it. So it can happen. It's just slower. And when we form glycogen, when we do glycogenesis and we store it, it actually gets stored in our skeletal muscles with a little bit of water. Sometimes it can actually make your muscles a little bigger. And so when we do glycogenolysis and we get rid of it, we actually are going to now lose water from our muscle tissue. When we lose that water from our muscle tissue, it goes to our kidney and we pee it out. And so when someone, if you don't eat for a day, you might notice you're like 4 or 5 pounds lighter. Well, when we say water weight, that's exactly what it is. It's the water that came out of your muscle as you burned up your glycogen. And then as soon as you eat and form your glycogen again, the water just goes back in. So we have these two processes of storing glycogen and breaking it down. And you might remember your hormones, insulin and glucagon from the pancreas, are antagonistic hormones, where insulin is anabolic. It's going to store glycogen, and glucagon is catabolic. It's going to break it down. But we also have gluconeogenesis. So this is when we make glucose from something that's not a carbohydrate. We can actually make glucose from glycerol, amino acids, and even lactic acid. So the main organ that does this is the liver. And so the liver is able to convert these products into glucose and release it. And you can see it's called gluconeogenesis. Again, we don't have to memorize the intermediary product, but the liver can take lactic acid, pyruvic acid, and turn it into glucose. It can take glycerol. So if your glucose levels are low and you've burned up all your glycogen, then you will start to burn fat and protein to convert into glucose, because we have to maintain our blood glucose levels. Red blood cells and neurons can only burn glucose for energy, so it's important that we are able to maintain those. If we look at the lipids, they're very similar. Remember that a triglyceride gets broken down into a glycerol and a fatty acid. They're very energy dense. So one glucose gives you around 32 ATP. One triglyceride gives you 460. So when it's broken down, glycerol just goes right into what we just did. It goes into glycolysis. It goes right through the citric acid cycle. Works just like a carb. In fact, remember, you can even convert it as carb. Your liver will convert it into that for storage. Fatty acids are different. Those fatty acids just go through a process called beta oxidation. A fatty acid is a long carbon chain, and it just splits it off 2 carbons at a time. And so beta oxidation, that is the process of a fatty acid being broken down into acetyl-CoA. And remember that acetyl-CoA is going to enter the citric acid cycle, and then it just goes through ox-phos. So when we're looking at our fatty acids, they're going directly into the citric acid cycle, whereas glycerol comes all the way through. When we have rapid lipolysis, so we're breaking down fats, right? Triglycerides to glycerol and fatty acids, lipolysis, breaking it down. When these come in and we're trying to break them down into glycerol and fatty acids, and you can see here the fatty acids do the beta oxidation into acetyl-CoA. There's my glycerol, can go into glycolysis and through. But what you'll notice is, look at the fatty acid arrow. See how the fatty acids only go one way only? They only go into acetyl-CoA, and then acetyl-CoA only has one place it can go. It's got to go through the citric acid cycle. Well, if you have a lot of acetyl-CoA and you can't get it all into the citric acid cycle, the only place it can go is the liver converts it into things called ketone bodies, because you can see that these reactions are irreversible. They don't go backwards. So if you're burning fat at a really high rate, so you're burning fat, you're breaking down, right? You have this lipolysis happening, and we're forming a lot of acetyl-CoA. Well, you would be burning fat if you probably weren't eating a lot of carb, because remember, our body's going to actually want to burn glucose for energy. So let's say you're on like a low carb diet, or you're diabetic and you're not managing it very well, so you can't really use glucose in your cells. Well, if you're not eating glucose and you're not using it well, you need glucose. And so this oxaloacetic acid, or oxaloacetate, it's the same thing. There's actually a whole separate pathway I'm not teaching you, but that gets converted to make glucose. It's one of the things that's going to happen if you're on a low carb diet. Or again, if you're diabetic, you don't have the insulin, so you can't get the glucose in your cell to use it. Well, when that happens and you decrease those levels, remember that first step of the citric acid cycle was acetyl-CoA came in and combined to form citric acid. So now if you decrease this cycle slows down. Well, as it slows down, you're still got fat coming in because that's what you're trying to burn. And so acetyl-CoA is going to accumulate. And the only place it can go is into the liver, where it makes it into those ketone bodies or keto acids, and this is called ketogenesis. Now, those ketones are going to leave the liver, and they're going to go to your kidney, where you're going to eliminate them in your urine. So you can do a little urine test and measure your ketones. The concern is something called ketoacidosis, or DKA, diabetic ketoacidosis, because if you're making the ketones faster than you can eliminate them, they accumulate in your blood and they're acids. So ketoacidosis is life-threatening. It can kill people. So if you're on a low-carb diet, or some people go on a ketogenic diet on purpose, you will lose weight. There's an optimal zone they want you in because you're going to burn fat for energy. The problem is they're very hard to sustain because remember, like vegetables and fruits are carbs, So if you're not eating many of them, it's also probably not the healthiest diet for you. But you really have to watch your ketones, because if they accumulate, and you go into ketoacidosis, it can be fatal. So again, it's usually with a low carb diet or diabetics, and it could also happen in a starvation diet because you're not eating. And these ketogenic diets have been used to control seizures. People with epilepsy, especially children, when they eat the really high fat diet, for some reason, they're not sure why, but it can help make the seizures go away. So that was lipolysis. Just a quick look at lipogenesis. We also need fatty acids and glycerol can be taken in by our cells and resynthesized into fat. So these are just some examples of some fats that we would need to possibly make, like steroid hormones, estrogen, testosterone. We want to have cholesterol as part of our cell membranes. We need the phospholipids for cell membranes. So lipogenesis is the building of fat for function, where lipolysis is breaking it down. And then with protein metabolism, we don't want to burn proteins to make ATP. We have other things we want to use them for. But if you're on a starvation diet, or you're not eating, or you're malnourished, if you don't have the glucose and the fat, you're going to use your proteins for ATP production. And so when we look at what happens for that, we'll look at this pathway. We have our proteins. They get broken down into amino acids. Amino acids have this NH2, an amino group. So what happens is you take that amino acid and you break that group off, and it gets converted into NH3, which is ammonia. And ammonia is very toxic. Your liver is going to, one of its jobs is to convert ammonia into urea, and it goes to your kidneys so you can pee it out. Now, if you're on a high protein diet, it's going to put more strain on your kidneys, because you have to get rid of all that urea. That's where a lot of it comes from. It's a nitrogenous waste. So sometimes people in renal failure, or they have kidney diseases, they might tell the patient to decrease their protein intake to save this pathway. Because every time you eat protein, this is what's happening. We got to get rid of those waste products. Now, once you take that amino group off, there's still a whole chunk of amino acid, and they call that a keto acid, not a ketone, a keto acid. And then just notice that can be used to make ATP as well. It can go in glycolysis or into the citric acid cycle. So we can burn protein for energy. So if we're trying to do kind of like a little summary of this, of the molecules, we know that glucose comes in and goes straight through, right? Goes through glycolysis, transition, citric acid. If we look at our triglycerides, they get broken down into the fatty acids and the glycerol. Well, remember, the glycerol just goes straight through, just like the glucose. The fatty acids, however, are going to go through that beta oxidation process, and they're going to go right into the citric acid cycle and through. And then if we're looking at our protein, I'll just put it down here, proteins are going to get broken down into amino acids. They have to get deaminated, so we're going to form our ammonia. NH2 becomes NH3, and urea, we'll deal with that. And then we have what's called the keto acid. That's just the rest of it. And then that can also enter and be used for ATP production. So because these pathways are kind of interconnected, we can take our glycerol and convert it back into carb or fat. Same with protein. If we take too much amino acids in, we can actually, those amino acids that come in, they can come in and be converted into glucose. So they can undergo gluconeogenesis and then be stored as glycogen. And they can also come in and be converted into fat. So that the pathways kind of interconnect. So too much protein, nowhere to store it. We're going to put it, it's going to be converted into glycogen, or it will be converted into fat. And then as far as anabolic processes, this is what we want to happen with our proteins. We want to bring them in. There is nowhere to store them. We are always turning over proteins. The essential amino acids are the ones we have to eat every day. So there's roughly 20 amino acids. You're not memorizing these, but maybe do a diet analysis and see where you're at. Because if you don't take in the amino acid, you can't make the protein. So the essentials are the ones that have to be eaten. If you hear non-essential, your liver can make them. And then why do we need protein? Well, I think you guys already know this, right? Enzymes are all proteins. We have transport proteins, hemoglobin, myoglobin, albumin, storage, ferritin. We didn't do a whole lot with storage proteins. Iron, basically how it can bind to iron. These are more like, I would think of them as transport proteins as well. Muscle proteins, structure, collagen, keratin, hormones, lots of protein hormones, and then everything we did in the immune system, all that protection. So this wraps up our lecture on the metabolic processes and the anabolic and catabolic processes of our nutrients.